Hello, everyone, and welcome to today's webinar, Nanoparticle Isolation via Nanoscale Sorting, Small Footprint, Small Particles, and Big Ideas. I am Cassie Soltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Beckman Coulter Life Sciences. To learn more, visit beckman.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I'd like to now welcome our speaker for today's webinar, John Tigges, Director of Flow Cytometry Core at Beth Israel Deaconess Medical Center. John, you may now begin your presentation.
Thank you, everyone, for joining today. Appreciate this opportunity to speak a little bit about work on the SRT and looking at nanoparticle isolation. This is something that has been happening for probably, 7 to 8 years now. Started with the work on the MoFlow XDP, moving to the Astrios EQ, and now what is hopefully, and you will see, a much more direct and easier approach due to just the, as we have here, that small footprint and the complementary status that is involved with the SRT to the actual rest of the cytoflex family, such as the S and LX. So we must start first with some disclaimers. Simply, this is a scientific presentation and is concerned with what happens in the laboratory here at Beth Israel Deaconess Medical Center. Also, I want to let you know that I act as a technical advisor and scientific consultant for Acarella. But again, this is completely scientific, non-promotional. So anything that is stated here is just basically what we've performed in a laboratory and not anything that is claimed by either company. So let's dive in. For those of you who may be here that have just started looking at nanoparticle analysis and more direct the actual nano flow cytometry, let's just a little bit of background, as we said, just to see how these things came about and why they're kind of important. So the most important aspect that we have going is this kind of, we started out with nomenclature. So when we talk about nanoparticles, and when I use these terms of nanoparticles, extracellular vesicles, you may hear exosomes and microparticles, microvesicles. They're all kind of interchangeably used. And so this is why the nomenclature became important from the very beginnings of just calling it dust from platelets and red blood cells. We have moved into this kind of small EV versus large EV. And this is just dictated, as you can tell, by size. So things that are in the 150 to 200 nanometer range and below are considered small, and above that considered large. And as we said, many things fit under these categories. So let's take a little further look on the importance here. So we have this whole thing of, okay, biogenesis, size diversity, can we load them? How do we get them to be very specific to a target for actual delivery of these contents or cargo? And how does this have any effect in, function in health and disease, or maybe as a immunotherapy, targeted therapy, diagnostic, prognostic? So they've gotten a lot of kind of push in this area, as this also encompasses everything that's going to be thought of as in, as you can see, viruses, the extracellular vesicles, the lipid nanoparticles, virus-like particles, viruses themselves. And so this starts to bring about these characterizations and how we differentiate these things. And so again, everything in nano flow cytometry comes down to sizing, and if we're able to see things based on our ability to detect using the systems. And so some are simpler than others, such as if you have apoptotic cells or apoptotic bodies or microvesicles. When these are kind of moving towards the larger end of nanoflow cytometry, most instruments can see this. It's as we start to creep down and get into this weeds of size or limited detection that we start to see a little bit more of the issue and how do we actually start to work around that. And so a lot of this talk is going to be ways to start to work around that limited detection, and also just understanding what it is and how to actually deal with it. So we're going to move on to looking at problems, right? We're going to start with a little bit as we go into similarity of the contaminants. So when we're talking about biofluids, we're having this question of what is actually in the soup. So as you can see, these extracellular vesicles share a lot of characteristics with apolipoproteins, chylomicrons. You can generate micelles, which have similar characteristics. If you have protein complexes, they all start to put into the same size and density of what you're actually looking at. And so isolation has become one of these major problems and kind of talking points, right? What is the proper way to isolate? And then again, we don't want to do too much manipulation. So is there a way to get around this issue using other technologies? And that's what we started to want to explore. Okay, so as we started to say on the previous slide, how there's contaminants and that's one of the issues, Houston, we have some problems. So things that we would be doing in cellular biology now have become more prevalent within this nanoflow cytometry. So while we consider a bunch of things when doing our cellular assays, they become a little more to the forefront because we can see more, right? Because we're actually dealing in a stage where we're moving down that threshold. We're looking at the bottom of the limit of detection, right? We really want to see where our signal to noise ratios are, what we can actually process. So things that may not have been right there in the front of our minds when setting up a methodology now become major issues. And just for a few examples, the all important saying, we are what we eat. And so this is something I've pointed out in previous talks, and just, that keeps coming to the forefront, in that depending on what you eat, we may see things that are representative of nanoparticles that are not. And you can see just from our actual lane changes here, or our intensities, certain markers for your extracellular vesicles change, certain markers for contaminants, such as the apolipoproteins, change. And so you can get kind of confused, or, if you're new to the field, you're unable to actually assess what is a particle of interest and what is not. And so you try to control as much as you can, and getting samples from, working with mice is always great because you can control the environment there. Getting patient samples that are like going through surgical procedures, as there is some also control there on fasting, or, when there's possibly blood draws, the timing, which brings us to the next problem, right? We have to be conscious of this collection also. Due to our circadian rhythm, right, the diurnal variation, there's a change in the actual aspects of what we see in time of day. So a lot of times when people are in some sort of procedure or in the hospital, right, they circumvent some of these problems, such as the needle size, because you may have the needle inserted, so you're not worried about that injury that happens right at the beginning. You're not worried about, changing of gauges or anything like that, or different, just how one person approaches it compared to another. Then, even more importantly, are the collection tubes. Each tube actually has a different aspect. And there was a paper that I was involved with, here a group led by Dr. Guerin, and a paper by Vivian Tran, that showed some of this using a multiplex assay to look at these extracellular vesicle populations within the different tubes that you could use. And you could just see, and this is basically there, not to go through the total interpretation of it, but just heat mapping, you can see how there are multiple changes within this, and all dictated by the tube. Now, a lot of this is not to just scare people and say, oh, don't do this, don't do that. It's more just to get you to think about it, for one, and to keep repeatability, right? So if you do something, do the same. You know, don't change between tubes. Don't try different needles. Don't try collecting at different times. Don't collect specimens from someone who just had a huge lunch compared to someone who is, you know, fasting for a blood test, or as an example. Other things which I'm getting into and may have slightly mentioned before, of things you didn't really have to consider in your cellular assays and doing your normal, or what they call conventional flow cytometry. So most conventional flow cytometry is based simply on Rayleigh scattering, right? And all your equations come from that. And so when you're going along and you're not changing thresholds too much, and you're not worried about how the light scatters in a different manner, things become pretty simplistic or conventional, and you can just keep going, right? The old plug and chug method. Whereas small particle detection is based on me scattering and me theory. And so the light of these is changed, right? It's shot off in different directions and has a different kind of scattering profile or dynamic. I like to think of this too in the sense of, you know, how these translucent particles, even things like labeling them, which we'll see, or, you know, how they go through, they're translucent, or how thick their membranes are. It's kind of like this mosaic, or you know, panes of glass, and how the light shines, you know, how you can see that. And all those things actually, when you start to move this limited detection down, become factors that we didn't think about. So getting into this refractive index problem, right, it starts to get you into this changes of nanoparticle size. And there's many different looks at this, from, you know, the masters at the me theory and refractive index kind of effects on these nanoparticles, such as Josh Welsh and Edwin Vanderpoel. I put down here just a little bit of literature on looking at the refractive index and how you can derive this from some of these characterizations that you're doing. But here we just wanted to give kind of a in-your-face example of how particles of the same size but having different characteristics or different refractive indices can actually skew some results. So if you just look at a normal polystyrene bead compared to a polystyrene bead that has, you know, a bright yellow-green fluorescence, and compare that to just a non-fluorescent, this case hydrogel, which has a very low refractive index, that closer to your background sheath and also closer to mimicking an actual biological particle. And you can see how you just get multiple profiles and you get different measurements. These are calculated through, you know, changing your arbitrary units to, you know, SI units. But also just simply, if you were looking at the MFI, you would have the same type profile. And being transparent here, and we'll see it in the next slide, I didn't change to factor in that refractive index, just for showing how that affects. So in the software, you can actually go in and put in the specific refractive index for these particles, and that'll change how they actually are represented when you change them to SI units. And we'll get a little more into how this standardization and calibration affects your data and actually makes it more robust. But this was for more shock and awe value of, if we're just looking at these particles and we overlay, you get a different profile for each, even though they're all the same size. And this can be even more exemplified when you look at this low refractive index 200 nanometer mimic compared to a 100 nanometer polystyrene bead. And so these are things, again, that we got to think about in how we actually set up our instruments and what kind of reference materials we use to start comparing and contrasting results. And this is just within one instrument. But imagine how this would actually start a domino type effect or become exponential when you're starting to do between instruments or between institutions. Here, I like to say that problems are solutions in disguise. I always like to say that thing, right? Things are not complicated, they're complex. And the major difference with that is when things are complicated, they're very hard to solve. And there's too many moving pieces and it gets confusing. Where if you just say they're complex, right, you can start to break it down into its smaller parts and understand it better. And this is what started to happen where more purpose-built instruments were there. You could find ways to change hardware settings in order to become a bit more centric to small particle detection. And again, I put in here that there's tons of protocols now that help us not make the same mistakes that others have, such as myself. I've made many. And I put here this little link for this small particle flow cytometry Discord that was created, where you can share protocols, you can share thoughts, you can ask questions, and you have some of the very, we'll call them experts in the field that are overseeing this or a part of this so that they can answer and share their knowledge. So this is a great resource if you're just starting. And even if you've been doing it for years as myself, I find this to be a great resource. I already had many different suggestions from that. So I'm going to give it a little plug here. But let's get into why any of this is important and how we were talking about SI units versus arbitrary units. If you've done any flow cytometry at all or any kind of measurements, most of these are arbitrary units and are not very quantitative. And so you had someone like Joshua Welsh and Jennifer Jones from the NIH, NCI, start to think about this in terms of how do we make the data more meaningful and how do we make the data actually a little more robust, right? So data assurance in this case. So they derived this thing called FCM pass. And so by using a set particles, in this case, you can use NIST traceable beads. They come with very well-defined characteristics such as nominal diameter, refractive index, and basically all the measurement standards there. And by running some of these along with possible fluorescence calibrations, you can put them through all the software and you get something that looks like this. This is twofold, something that I have really relied on to understand the performance of my instrument, to be able to calibrate correctly, and to be able to compare results over these longitudinal type studies, or when we're working with another institution to really get a project together. So you can find where things may have gone wrong and really talk to your engineer, and it gives them some idea of where things may have gone wrong. And so it's a freeware. I suggest, using something like this. Like I mentioned, Edward Vanderpool before, he does have also a very similar thing, all based on this Mies theory and the scattering properties of the particles. But let's get into what we all came here for, right? So let's step it up a notch and move into the actual sorting of particles. So the Cytoflex SRT, the reason we started to try these projects on it is was deemed and is still deemed the Cytoflex that sorts. So if we could do certain things on our Cytoflex S and LX series, well, why couldn't we just translate those over to the SRT? And as stated right at the beginning of this, I had always been trying to use sorters to isolate particles. So I had the methodologies already in place. I had a system now that was very similar to things I was doing for, our acquisition aspects. And so why not try it? So here is actually what we did. We took the same thought process that was involved with our Cytoflex S. We changed our 450 filter to the violet side scatter, or if we needed the 450 for certain aspects, then we could change that with any of the others in the violet pod, as these are pretty much plug and play on the Cytoflex. Just the easiest one to state is just changing those first two around so that you have a violet side scatter channel at your disposal. We then went into these NIST bead sets and calibrations using also the FITSI MESF beads, and cross-calibrating with 8 peaks, and then we use some yellow-green fluorospheres in order to also get more of an aspect of that fluorescence part, as NIST beads are not labeled. So our first step, we went ahead and sorted some beads. So as we just mentioned, we used our yellow-green beads of different sizes. We mixed them, put them into the SRT, and just put some gates and isolate it. Now, the reason that this is here and it seems very simplistic is, well, that's the whole point. We want it to be simplistic because if we can't sort the simple things, then going on was pretty useless. And, you've got to start somewhere. And obviously it worked, and it worked well, as you can A, see, and B, this talk would be very short otherwise. So, but this is useless, right? I'm going to admit that. It is very simple. People would say, well, sorting beads. Yes, I understand that. And I've said this, and I will restate it over and over again. Beads are beads, and we want to move on to a little more complexity. So we started looking at our recombinant EVs, the EV reference material. These are now commercially available through Sigma Aldrich. They were created at Dr. Hendricks's lab and are very well characterized. Hence, they are known as recombinant EVs and are a great mimic. So knowing the size, having an intrinsic fluorescence with the GFP, and many other characteristics that are similar. Now, we did not use them all. You have them at your disposal to make them a little more biologically significant to what your project is. But for our proof of concept, we just wanted to use that GFP to understand our machine and start putting together some protocols. So we kept it simple again. And basically here, we're going to start looking at this. We have our pre-sort listed above. We looked at our GFP positives. We then sorted those, as you can see on the bottom, to pretty good efficiency. And then we looked at the MESF values according to how we calibrated here and added those in. And then also for sizing. And so basically what we just wanted to show here is that as your pre-sort characteristics and your post-sort characteristics stayed very in tune. So there was not major changes. As we know, sometimes sorting may cause changes. And so we just wanted to make sure there was nothing we were doing during the sorting process that actually had an effect on these very well-known sample. So you can see here our equivocal GFP molecules pretty much stayed in line, and our sizing was relatively the same. We also wanted to see, and we're going to get into this, dig a little deeper to this later, right? We're going to get into it more. And so what we have here is fluorescence triggering, right? We have this at our disposal, and we want to see, well, what's the difference, right? And so we just did the same simple experiment, adding a fluorescence trigger, and we saw here again, size remained constant between the two, the pre-sort and the post-sort, along with the median FITSI intensity, we'll call it. And so, but we did notice one thing, so you can see that in the table here, is when we look at violet side scatter versus fluorescence, our median fluorescence intensity does not change, right? We have the same MESF value, relatively speaking. The percentage difference is not anything significant, but our nominal diameter changes. So we find that when we looked at fluorescence, we get a better limited detector, right? It increased our limited detection, basically because we're able to actually see a little bit more. We're able to go down and move the threshold needle lower in this instance. And so we wanted to think about this, right? Kind of put it in the back of our minds and say, that's interesting, but I think we need to look at this a little more. And so we kind of said, put it in the back of your head, think about it, and decide what we wanted to do. And so what we did before that is go back to kind of, okay, well, we can do these just beads. We can do the fluorescence based on the recombinant EVs. But what if we just start mixing a bunch of things that have, different fluorescence intensities and different sizes and different refractive indices? And can we just separate those? Again, keeping it simple and kind of, as I like to say, we set the bar a little bit low so our chances of success were better, as we just don't want to have an absolute failure. And you can see here, not much issue, even though, our picture there all the way to the left that has all the populations is not necessarily the most beautiful. But we were able to put these gates here, use our fluorescence to kind of separate and pull out multiple populations. But now we want to get more biological. So let's go into our bag and see what we have that will mimic those nanoparticles that we want to actually do for research samples. And so here's just kind of a list of the things that we did. We had some MLVs that were given to us. This was a commercially available product previously from VeroFlow, and company that was out of the University of Ottawa, partly with, you may know, Vera Tang. And these, again, very well characterized, very well known. And so they were a great mimic for our experiments. Unfortunately, they're not available anymore, as they were a lovely product. And I think if you just maybe find yourself a virologist, you may be able to get your hands on a similar product. We also purchased from Acarella some ACO-RL liposomes. These are reference liposomes, obviously, and again, well characterized with intrinsic fluorescence. Hence, we can actually understand everything we need, the concentrations, the diameter, and the fluorescence intensity. So very good product to kind of push forward. These are commercially available. We have a colleague and collaborator from Boston University, Dr. Gumul Luru, and he actually has these virus-like particles that are generated very similarly to the recombinant EVs, but we were able to get these with both a GFP and TD tomato fluorescence, which added just a little bit more to what we were able to try with the sorting samples. Again, all well characterized, all well known, and so it allows us to know what kind of effects the sorting would have, and also gives us the best chance to be successful. So the first thing we did is we took some of our MLVs and we also took our VLPs, which have the GFP or the 525 fluorescence, and we were able to take a look on, could we just sort based on epitope density? So as we go into the next one, first thing I want to point out is these gates are arbitrary. There was really no predefined or notion of where they should be. We just arbitrarily picked these spots and decided, could we separate when we had this just kind of heterogeneous push of fluorescence, right? There's not well-defined aspects here. And as you can see, and we actually were very shocked and surprised that it did work, as we were being so just kind of open with the gates and, oh, this is low and this is high and this is medium. So as you can see there, we're able to separate out in multiple intensities from the two products, and started thinking, okay, well, it is possible to start just sorting based on this epitope density kind of notion. And so that was a great thing to think about and a great thing to have as part of the capabilities of the SRT. It kind of, again, was very shocking, and that we were able to have that kind of precise sorting, and that, we thought there'd be a lot more overlap. Now, what I want you to keep in mind too as we go along is how these post-sorts start looking. The post-sorts, when you start trying to separate things, do show the aspect of the separation, but you're going to notice too that the percentages may not be what we used to when we're looking at our normal cell sorting, right? We're always looking for that 90% or better, of our sorted population. We're trying to do purity or single and get really, really high purity results. And that's not exactly what we're seeing here. And so, again, another thing I'm asking you to put into the back of your brain for a moment that we're going to touch a little more base on. So we did start before, and we're going to look a little bit more, as we mentioned, to which is better, a violet side scatter or fluorescence thresholding. So first things first is just let's get a normal kind of look at, if we just set it up on the cytometer, how does it look? And you can see violet side scatter here versus fluorescence. We have the same thing that was mentioned earlier. Our fluorescence remains constant, yet our diameter changes. And so we took those, like I said, because we had the two different fluorescent proteins on these, we started and said, okay, let's go with some violet side scatter and let's mix them. So we mixed them, we separated them during sorting, and we get these two populations. And like I said, put that in the back of your head of how the numbers aren't great, but there's obvious separation of the two populations. And so we have this kind of skewing of how well we did, because our percentages don't convince us, but our visual does. And so we're seeing what we want to. It just doesn't make a lot of sense. And so we're going to start talking a little bit about noise and how noise affects these things. However, we don't have to worry as much if we go to fluorescence, because we can really just move the threshold in. And because we're only looking at things that are going to be positive for the fluorescence, and in this case, that dual fluorescence triggering, we get tomato, GFPs, and we separate them, right? Very clear separation, very nice. And because we're only looking in those samples of something that has fluorescence, we're able to see very nice positivity. We're getting the numbers that we think we should be seeing. So the background's decreased, or that noise that is there. We're also not too worried about the refractive index of the samples, right? This starts explaining a little bit on why we're seeing a different nominal diameters, because fluorescence is agnostic of a refractive index. And so things become very focused on just the positivity and not necessarily what is happening in the scatter. But yes, it is problematic if you don't have fluorescence. So we have to still think about all this, right? And we have to think, okay, we're lowering the limit of detection based on actual looking at fluorescence. And we said it doesn't change, though, what your intensity is. It only changes in how the threshold is accepted and how the limit of detection is affected on the instrument. So it has its benefits. So we went a little bit further because we said, okay, we can't just accept that. We have to keep going, and we want even more. So we had a colleague come in for a demonstration of the SRT, sorting these fragmented Ebola viruses, non-infectious. And you can see again, separation is great. Numbers are terrible. So moving again to, okay, fluorescence. So this is just really to hammer home how it's all visual, right? It's how we've been pre-programmed to look at success. We look at success on, did we increase the population? Did we enhance what we were supposed to sort? And in the, when you look at numbers, when you say, well, the other ones were only like 1%, 2% or 10%. Now we've got 80%. So this is much more successful. Again, we're just blocking a lot of the things that end up. And so it's not a factor that violet side scatter is not a useful tool. It's a factor that we become concentration dictates everything. So we're going to get into a little bit more of things are a bit noisy, just to get into this dark noise and how we circumvent some of these problems. So you can see here what we decided to do next is take our violet side scatter threshold and look at the post-analysis, whether it be on the violet side scatter trigger or the fluorescence trigger. So we sorted on scattering and we looked at it pre-sort one way, post-sort both ways. So here you can see violet side scatter triggering, still same low numbers, but we were able to isolate and enhance. And then you get the same details if you just go to fluorescence triggering, which shows these high percentages. So yeah, it's a little bit of a misconception or smoke and mirrors at this point, because if you sort on fluorescence and then you look at those two, so again, using pre-sort one type thresholding, and then we analyze using both thresholds, and you can see again the numbers, if you just looked at the top portion, don't make sense, right? Why are they basically going lower? And again, this is simply a factor of concentration. As we sort, we're actually diluting the sample a multitude of time considering the amount of particles. So you can concentrate them. That will give a little bit better because, as I said, concentration issue. It's a signal to noise ratio. As signal decreases, noise increases, because the noise is a constant value and where it's positioned. So the only thing that can change that is number of particles. So obviously, if you were to concentrate these and look at them on the scatter, you're probably going to enhance that signal and you're going to get a better perception of what's going on. The fluorescence trigger is agnostic of that because you're only looking for things that are positive. You don't get that real heavy background noise. So it is kind of just a workaround so that visually and numbers make sense. Hopefully that does make sense to everyone, because it is not the easiest concept to think about when you're thinking about limits of detection and this background dark noise that is created by the instrument itself from the APDs, the heat dissipation from them, and also just how it's having to produce that signal. And so you get just a lot of background, right? Like I said, totally different than when we do conventional flow cytometry, because we're not worried about the lower end, so we never see it. Why not both? So if we just start playing with these things and we add them together, right? So we can get, if they have fluorescence, great. If they don't have fluorescence, well, we are also accounting for that. Now there is a bit here of least common denominator, right? So the lower the amount, it usually dictates what's going on. And so in this case, fluorescence does have a little more influence when you do this dual thresholding. And so that you are actually probably skewing slightly towards the fluorescence aspect compared to the scatter. But if they don't have any fluorescence, you at least have a backup plan. So you can see here, we started looking at the liposomes with the recombinant EVs, started mixing these things. We're going to mix a lot of stuff together just to try to separate it. Here we mixed them together and we tried to isolate our liposomes by themselves. And again, just putting numbers there so that we get an idea of no effect on actually what is happening within the sorter to the particles, and that we are getting these high numbers and seeing some of that background also. So we're not ignoring the noise, we're just diminishing its effect on our total data. Here too is we started to notice this thing within the recombinant EVs and also our virus-like particles, where you had negatives and positive of the GFP. And we started thinking, was this just noise or was it an actual GFP negative portion? And so we sorted them. Why not, right? This is the whole point of all of this, is to show that we could sort and maybe garner information from these subpopulations that we didn't need before or we didn't know about before, and just to advance the science, right? I mean, some of this is just us having fun, right? And trying to understand better. So you can see here, we can isolate the two, and then it hit us. Well, okay, maybe it didn't hit us. We started talking to people as we had some idea, but we got it confirmed. So if you're actually putting this and you're trying to generate these virus-like particles by adding your virus, plasmid into the HEC293 T cells to generate these VLPs or recombinant EVs, well, the HEC293 T cells are also generating their own EVs. Hence, we have the GFP labeled from our constructs, And we have the unlabeled EVs that are just from the HEC293Ts. So again, something very simplistic that you could kind of put the logic to, but was very well seen and understood through the sorting methodology and by isolating them, right? And understanding what was actually happening, the biology. And so you had these two different levels, and now you had them isolated, can do different things with them. Also, in these instances, it shows a little bit of what we were talking about with problems. Contaminants, right? These two share density, share size. You can't separate them. You can get rid of the cellular debris and other things that may happen in these cultures, but you can't get rid of the two things that are very, very, very similar in all their characteristics. All right, so biofluids, right? It's why we want to be here. We want biologicals. So we can test commercial products, we can test things that people have made and characterized in their lab, but we actually want to do some real stuff, right? We want to get on to the science of it all and how we actually narrow down and what this is useful for. So we took some plasma. Here's some simple things, and we, how it was prepared, cell-free plasma, or some you can use, platelet poor plasma. There's many different aspects in this. And we performed some spike-ins just to start thinking about this. And then we started thinking about labeling extracellular vesicles in order to, or lipid membranes in this case, in order to actually see what we could accomplish. So the first thing we're going to do is we're going to take some plasma, we're going to spike in the liposomes. We spike in the liposomes in the two, and then we sort them out of the plasma. So as we know, plasma is messy. This is just normal healthy donors, self-proclaimed normal healthy donors, as it's listed always in our papers. And basically with the spike in proved, okay, if we had something with fluorescence in a kind of murky, not great substance such as plasma, can we get them out? So first things first, spike in, overabundance, pull them out. Here is just basically what we did with the dye and what the dye is. This is ACO 490 COE from Acarella, another commercial product that has a fluorogenic property. It's hence why we thought it would be best as in a solution. It does not light up. It will only light up when it transects the bilayer of these particles, such as extracellular vesicles or viruses, VLPs, bacteria, and things of that nature. So we went with this to try to circumvent some of the issues that may occur if they're leaky and or just appear within solution. We took our revs, we then spiked those in. So the recombinant EVs went into our plasma samples. These were pre-labeled with the ACO 490. And for some reason, when we looked at these in general, they showed a much clearer kind of double population. I didn't show it here. But when we put them into the plasma, they kind of had this almost, is it double positive or single positive? We didn't get overly concerned with it as we, for this type experiment, we were just trying to throw in and fish back out. And as you can see here, we then took the sorted sample that we saw previously there, the bottom portion, that is that 83%. And we brought this to the microscope to perform Dock field imaging. And so what we did on the recombinant EVs there that were GFP and ACO 490 is to look for those that were single labeled or double labeled. This is obviously just a small portion of the sample. Basically, we took our sorted sample and it's 10 microliters on a slide and imaged those. But we were happy with not just seeing that it was on a post-sort, 83%, but that we were able to image and understand that our sorting was successful, right? The orthogonal methods in order to prove that what you're doing is correct. We went one step further and we said, well, that's great. We can actually do labeling of something, put it in, and then get it back out. Well, what if we were to take something that was unlabeled, put it in, and then add a dye to see what we can pull back out? So that's what we did. We took our red blood cell EVs, we spiked those into plasma, And we tried to do it in a low amount. So we basically took what is, 1 times 10 to the fifth, basically 100,000 red blood cell EVs. And then we added that into our plasma that was basically diluted a 1 to 1,000. And for those not familiar, that is not a very large dilution of plasma. Usually when we're working with plasma, we get into the dilution of 1 to a million. This could have simply some effect too with how it appears to the machine if you don't really get rid of the yellow tinge that is associated with plasma, that kind of yellow orange that occurs, because the instrument will pick up, as we showed before with the refractive index and that whole stained glass kind of approach of how any kind of change to that will change the refractive index and maybe your scatter. We did test this before, and we were able, obviously, to get a decent figure here on the left of how the RBC EVs looked in the plasma. Our pre-sort, so we have the first picture is basically plasma alone. Second one shows the RBC EVs in the plasma, and then our post-sort of that. And I'm going to own it. As you can see in the next couple of slides, it does say PLAMSA on the top, because I'm hopeful that my sorting technique of nanoparticles is a lot better than my ability to type. So I will firmly own my inability to hit keys properly, and I guess even more appropriately, my ability to recognize errors. Someone else pointed out to me, and I said, well, I'm just going to leave it and state this little fact that I guess I'm a better cytometrist than linguist. But back to the actual science, you can see there the pre of the fluorescence for the ACO 490 and the post. And to make this even a little more efficient and bring it home is we then just took cell-free plasma, labeled it with ACO 490, no spike-ins. We then looked at the post-sort, which we did do take some and put it through the Amicon. So at first we removed any excess dye with some Captic Core 700 beads. Then after sorting, we used an Amicon filter to concentrate so that we could get this lovely picture of the sorted particles, and also to look at the fluorescence and enhancement. And once again, we took these down to our microscope and did the same process of dark field imaging and fluorescence overlay to show all the labeled particles that we were able to isolate and concentrate. So this was a huge success, and why we went through the last 45 minutes of discussion all to get here, to actually be able to look inside plasma and sort out positive sample. Granted, am I saying these are all just extracellular vesicles? No. There's a good chance they are, simply because we are using the fluorogenic dye that needs to transect the bilayer of an extracellular vesicle, so it should not label lipoproteins that may exist or other things. These samples are cleared, hopefully, for that task. And on top of that, we do get a nice picture. We're not seeing a lot of other large contaminants or clustering. So we had some confidence, and it's, this is just one representative, but it's not an N of one. So we have been able to do this a few times. But, if we were able to do that, we're never satisfied, right? No one's ever satisfied. So what's inside? Can we even go further? So this is something that has been going on with the lab of Dr. Guerin, with the help of Dr. Pereira. And so basically what they did is they created this loop sequence, right? This is a hairpin loop molecular beacon to the lab of Sanjay Tyagi and Rutgers. You can get these similar from companies like IDT. And so you build your identification of a target. They actually have a build sequencing kind of program within their website so that you can type in. It will build you a reverse transcripted molecular beacon sequence with a fluorescence reporter and a quencher. So basically in the hair loop, as they're close together, you will get zero fluorescence. As the target binds and it separates, the reporter is able to fluoresce and therefore get a signal. And this shows the presence of the particular beacons. So to make life a little bit easier, because, well, microRNA are very small. So we didn't want to totally just go for doing this. We found it gets very difficult. So the easiest thing to do is we tried first to get a streptavidin coated bead to attach on the biotinylated molecular beacon. And then we just added targets for the microRNA 451. And we put those on the sorter. And so as you can see here, we have our bead population that we were able to isolate, make sure that we did not have any on the far left there, make sure there was no kind of duplicates presented. And then we looked for the beads specifically and then looked at them because they do have, as we said, a reporter, which is this fluorescence, fluorescein, FAM, also known, and then just sorted on that FAM positive or the fluorescence reporter. But again, beads, targets, got a test, but it's not what's really interesting, right? It's not the great sexy portion of how to do these things. So then what we did is we took our plasma once again, and we added our beads that had the biotinylated microRNA 451, added that to the plasma to see, could we pick up free microRNA within the plasma and sort it. So as you can see, we have the beads that we located. It looks messy, I will admit, because again, plasma, not the greatest thing to work with in this type scenario. We then looked at the fluorescence, was it available? And then we sorted it. And as you can see, we started to really isolate the bead population there on the upper right. And we were able to see just the fluorescence of the actual reporter, meaning it bound the free microRNA 451A, as it is. Molecular beacons are built to be very specific. And again, we just didn't want to take our word for it. And I apologize if the image is not great. It looks so much better when it's not here in the webinar, but I guess we can take home the specific message, right? That we have without the targets, right? Just the plasma without actually putting on the molecular beacon itself, we don't get anything. So these are the 200 nanometer beads alone without the actual ability to fluoresce. And then we have all these little blotches as I see them on my end. Hopefully they come up better on your end, that exist, that are green from the FAM. And we get to the finality, right? The final chapter. And I call it sharing is caring, because when we go out in the wild, not all our SRTs are made equally. You know, while they sit in the same platform, they're not alike. And so this is why that standardization is so important for rigor, reproducibility, and to be able to share data and really have quality assurance. So again, with the FCM pass, allowed us to take our instrument at Beth Israel Deaconess and to compare it to a lot of work we did with University College Dublin with Alfonso Blanco and his student there at the time, master's student at Annie Casterson. And then we were able to work with her and Alfonso and my colleague, Vanessa Costa, to compare and contrast results. And by doing this standardization, we were able to make these kind of samples. And there's a lot more, obviously, that we're not showing at the moment to show these whole collaboration that we did. And hopefully we'll be able to show that in some sort of manuscript soon. And here we're just showing that, again, if we look at our numbers, if we look at the MFI, very comparable. If we look at what we have for both fluorescence and scatter, we're getting similar numbers. And if we look at the actual SI type units, our MASFs are the same and our sizing is the same. And this just proves that by using standardization, we can really start doing very collaborative science and make things go along a lot quicker, right? More hands without actually having them all in the same place. So as conclusions, nano flow cytometry is a very complex methodology. This has been built on for years and years and years, and has become not only about the instruments, but building an actual protocol and method that works. And like I stated, with the Discord channel and a lot of other YouTube videos and protocols online, there's a lot that has been done and the mistakes have been made. And we have put together things such as, you can look at the MySev guidelines, you can look at MyFlowCyte EV guidelines, the compendiums, just to get a better idea so you don't have to have the same struggles. Isolation of samples is essential for that downstream analysis. And here we've put the possibility of sorting methodologies as an isolation method. And nanoflow cytometry offers this method to analyze and isolate those nanoparticles. The Cytoflex SRT is shown to be effective here as an instrument for flow cytometry, and it's piggybacking on the rest of the Cytoflex family and the ability to use these violet side scatter quick switch to really enhance the limit of detection and allow you to get better data. And again, to circumvent the issues related possible to inherent scatter noise, you can use fluorescence thresholding or the dual thresholding so that you are able to see fluorescence and non-fluorescent particles. Of course, acknowledgements. I would be remiss to not thank Enid Seguiron, who not only does the microscopy for me, but anytime I have any questions about anything in the nanoparticle world, he seems to have an answer, or someone I can talk to that will have an answer. Dr. Pereira, who has gone back to his home country of Brazil, we miss him and his ability to really work with nanoparticles, especially red blood cell EVs. Vivian Tran, who helped with the experiments, and as I mentioned, Vanessa and Rennie. Rennie is now in the lab with us and continuing on this science and really trying to perfect the art of sorting of nanovesicles. The NIH, Jennifer has been a great mentor, helps me understand sorting. I was able to work with her with many different aspects of sorting throughout the years and really learn the biology and biogenesis behind what I was trying to accomplish. So I thank you very much for your time. I hope that we have hit the major aspects and that this gets you excited to go out and try some experiments on your own, or find someone with a sorter and convince them that this is possible and that the experiments can be rewarding.
Thank you for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just type your question into the Ask a Question box and click Submit. We'll answer as many of your questions as we have time for today. Let's go ahead and get started. Our first question asks, what modification to workflows were necessary for the Cytoflex SRT to perform nano flow cytometry, such as background instrument settings and swarming?
So this question actually was touched upon a little bit, and I'll try to add a little bit more to it. The issue being is it is quite extensive. But for the SRT, we did similar to what you would do for Cytoflex S that has been put into the literature. It's a simple change for the optical bench of changing the 450 filter with a 405 filter. So you just kind of swap the locations. For swarming effects, we use just a serial dilution to make sure that we are getting a consistent and representative population of the cells. So by doing the dilution and making sure that you keep your speeds at a lower rate, somewhere in 1,000 events per second, 2,000 events per second, you don't want to be worried too much by going high into, things like 9,000, 10,000 K. Then you get a lot more aborts and you could open yourself to that swarming effect. You can also cause changes in your background. I know that was part of the question, I believe. So having that and actually putting in a PBS to make sure that your background is sufficient, that you've threshold properly, and that you're giving yourself best chance to see the lowest possible size of a particle, a nominal diameter. Additionally, we mentioned using fluorescence as a threshold or trigger. This will eliminate some of these issues also, just by removing some of that background that's associated with a scatter property.
All right, excellent. Thank you. Our next question here asks, what troubleshooting steps would you take if your sorted EV populations show poor purity or yield?
So it does go back to what the first question kind of touched on too, is making sure that you have not a lot of background, that you have a very clean instrument. This is just a normal practice within cell sorting when doing post sorts, is to just have clean in between so that you can get rid of any residuals or any other debris contaminants that may have been involved. And as we had mentioned in the talk, is that it does become, right? You get the poor yield or purity due to a concentration issue. So for us, when you really want to get those numbers to look as if you were doing a cellular biology sample and not looking at all the background noise, is keeping the concentration as high as possible. And this can be done by sorting a lot more particles or doing some sort of concentration step of the sorted sample.
Wonderful, thank you. Our next question here says, in a high throughput environment, how would you scale up sortings of EVs for downstream omics applications, e.g. proteomics and RNA-seq?
So this is an interesting question that I get asked a lot in the lab, and most people do not like the answer. So it is a numbers game. So you need a lot of beginning sample in order to make sure that you have a sufficient amount of yield in which to do downstream applications such as omics. So we have done this in the past with collaborators, and we have actually sorted upwards of 72 hours to make sure we had enough product to come down the line for the multitude of omics, like proteomics or transcriptomics or any of these other kind of assays, looking, microarrays, things of that nature. So it is a long, tedious process, and obviously some of it can be circumvented by having very positive samples. So if you're looking for some sort of construct or a particular surface protein, then if there's an abundance, then obviously, just like any other sort, the time goes down because you can get a lot more material from the positivity. But most of these are very low, and it's just a matter of time.
Wonderful, thank you. And it looks like we have time for one more question here. So our last question asks, could you discuss the implications of standardizing nanoflow cytometry data for collaborative research efforts? And do you think this makes replication of data and collaboration easier?
So this is a very big topic. It actually has its own working groups and task force within, ICEV and ISAC, flow cytometry and EV science. And so, by using these things, like we said, of FCM Pass or Rosetta or any other maybe self-made program that someone comes up with, getting things out of those arbitrary units and moving standardized units really helps to, make it so that you can do instrument to instrument comparison and institutional comparisons, and even work with colleagues, Australia, Japan, Europe, everywhere, right? So there are papers about it. There's, you can see how these things are actually done. And this has been a major focus even with the instrumentation. It was one of the big things that, we were shown in the development of the Cytoflex Nano, to have that kind of built into the QC and the products that they put together with it, so that one nano and compared to another would give very similar results. And this is all done through standardization and moving to actual standardized units, and not just kind of these what we're used to in flow of, this one's bigger than this one's more than that, by actually giving them specific values and having it become quantitative instead of just qualitative.
Wonderful. Thank you again for your time today and your important research. We would also like to thank LabRoots and our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.